Favorite 2-minute papers episode?
Oh, AlphaFold, it's easy. It almost felt too easy. It felt like too many of ideas were working. It was going up, and I remember talking to Tim, the engineering lead, going, this is really feeling too easy. We're having too much success. This problem can't be this easy. Are we leaking the test set, right? You know, are we doing the classic machine learning sin?
Fellow scholars, I don't really like to be on camera, but there is a big reason I am here today. You see, I met Nobel Prize-winning chemist John Jumper last year, and we talked for an hour. And in that hour, I learned more than I thought I would learn in a year. It was unbelievable. And today, I have the opportunity to give you this amazing gift too. So with that said, hey John.
Hello. Oh, goodness.
Really grateful to have you here today. I have goosebumps, which I have carefully hidden under this lab coat. So, what is AlphaFold and why is it important?
So, AlphaFold is a neural network, which makes it relatively appropriate for the podcast. But it is a deep learning system that predicts the result of a specific scientific experiment. And to tell you about that, I should tell you about the domain that it's in, proteins. So proteins are the nanomachines that basically drive your cell, a couple thousand atoms each. They're coded for by your DNA. When we say that DNA is an instruction manual for the cell, a lot of what it's telling you is how and when to build proteins. And so 3 letters of your DNA map to individual one of 20 chemical groups. Those chemical groups are basically just like little collections of atoms, you know, boop, 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 boop. And there's a machine, another protein in the body, that reads the DNA in a relatively complicated process and kind of builds out the proteins one step at a time, joining links in a chain or a rope. So it takes this chemical group, attaches that one, attaches that one, attaches that one, attaches it basically the same way each time, and builds out a string of maybe 300 of these as a reasonably typical length. And then what happens when your cell builds this thing is, of course, it's not a machine. Most of them are not machines that function kind of just as floppy ropes. They actually, some parts of it are greasy, some parts of it are positively charged, some parts are negative. So it will fold up, it will make helices, it will make sheets, it will pack into a relatively compact 3D object that is kind of the assembled working machine. So these are machines that build themselves, right, are joined in 1D. And of course, our DNA is 1D and our world is 3D. So this is kind of how the body solves this. It builds these things, they fold up into this incredibly intricate shape. And this happens, you know, there are about 20,000 human proteins. There are hundreds of millions, billions known proteins across all organisms. And one part of what I described is really, really easy to measure. It's really easy to read our DNA thanks to the genomics revolution. You can think of it as pennies to read the sequence of a protein, the DNA that becomes the linked amino acids. It takes a year to get the structure of a protein, and really hard experiments, and they often fail, and just extraordinarily difficult. If you want to put an economic value on it, maybe $100,000. So scientists do this experiment where they start from DNA, but they really want to understand how this machine works, so they need to see a picture of it. And so they determine the structure experimentally. They use enormous synchrotrons the size of small villages in order to do this. And so, but people have done it a lot. There's been an enormous societal investment because it's really important to understand this, to understand disease, to do drug development. There are about now 200,000 known protein structures, about 140,000 when we did AlphaFold. And we developed a deep learning system that goes from amino acid sequence, DNA sequence, to the structure of a protein in 5 or 10 minutes instead of a year, and does this with accuracy close to, not quite as good, but very close to experimental accuracy. And it's been used enormously. So we've predicted the structure of about 200 million proteins, every protein from an organism whose full genome has been sequenced. Scientists are using it for drug development, to understand the body, everything else. And I think from a machine learning point of view, it's both kind of the first problem really, really transformed by AI. It's an extraordinarily practical system that scientists are using. I think it's something like 3 million scientists have used our database of predictions. People make predictions every day with this. And it's also this kind of promise that we're going to use AI not just to do things that humans can do or solve human problems, but to do kind of superhuman level, there are no humans that are good at getting the structure of a protein by eye. They do it with experiment. That we can use this to transform science, that we can build new tools that fundamentally advance our science.
Now, I remember asking you last year, how did it feel when it first started working?
The time I really remember is that when it first started, or really what would happen is we kept, AlphaFold is built iteratively. It's not yesterday we didn't have AlphaFold, today we did. It was maybe 2 years and probably 30, 40 different kind of individual ideas that worked along the way. Some grand ideas, some small ideas, but each one kind of inching up the performance. And I remember maybe a year into building AlphaFold 2, the one that was really very successful, it almost felt too easy. It felt like too many of our ideas were working. It felt, it was going up. And I remember talking to Tim, the engineering lead, going, this is really feeling too easy. We're having too much success. This problem can't be this easy. Are we leaking the test set, right? Are we doing the classic machine learning sin? And he was sitting there going, I don't think we are. And we went back, we double checked, we zeroed coordinates in our eval set to make sure we weren't actually leaking. We couldn't really ever find a leak, but it felt too easy. It felt like the nature shouldn't yield this easily to our efforts. And I remember I wasn't really totally sure until actually we did some structure predictions for SARS-CoV-2 proteins related to COVID. And then the experiment came out afterwards that we were really sure that, okay, we were really not leaking anything. But it was wild.
Wow, that's crazy. But that is also the hallmark of a pro scientist, because during a research project, you miss 1,000 balls. And when you finally hit one, you don't ask questions. You celebrate. But that's not what you did. You picked apart the performance immediately instead. So that's amazing.
I mean, but you see pro athletes, they don't, they're when they miss, they're always interrogating, fixing, thinking. This is a craft. Machine learning is a craft, and you have to be a craftsperson to do it.
All right. Now the score. I'm asking this because the score didn't jump from 0 to 100 in just one magic trick. So this was a sum of many brilliant little puzzle pieces, and each of these contribute a little to the score. You add another puzzle piece, you get another few points. And what I'm wondering is that this sounds like a linear progress. You're climbing step by step. So why is it so surprising that when you get to the peak?
So, much like Moore's law was a succession of ideas and breakthroughs that in total gave the appearance of inevitability. And that inevitability in the case of Moore's law was driven by, exponential growth and investment as well. When you do this, you never know if you're going to get the next win, right? You don't know. And in fact, we have charts of progress, and they don't actually go like this. Right, the ideas that we list maybe go like that, but the actual progress went flat, flat, flat. Oh, what about this? Idea, flat, 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 flat. Idea. And in fact, the flat versus up. At the time, DeepMind was kind of on 6-month cycles. So every 6 months, you kind of formally continued your project and you presented your results to the whole company every 6 months. And I remember the first 3 months, we would always try our wildest ideas, and it would mostly not work, and we would get very scared, and then. About halfway through, we'd be like, okay, guys, we got to get serious. We need to not have no progress. And then suddenly some idea would hit, and then a bunch of ideas would hit. So it was always, it was alternating elation and terror. It's more, it's only when you make it really blurry and you squint and you zoom out, oh, it went up linearly.
Yeah, it's like overnight successes, 10 years in the making, right?
Yeah, it's that sort of thing.
All right. Can you build an intuition on what proteins would look like when folded up into a 3D structure? And also, did you have a protein structure where you looked at the 3D result and said, that cannot be right? And it turned out to be right. Does that happen?
Okay. I'll tell 2 stories on this. I mean, an intuition, you mean, can I build an intuition not on an individual protein? So sometimes you can say, oh, this looks really similar to this other protein. And therefore, I bet it's going to have about similar structures. So that's like what humans can do. And people call it homology modeling, which is a very fancy name for saying, well, the sequence is similar, probably the structure is similar. So you can do that. And sometimes you can notice individual motifs. Like there were all these papers that would list all these motifs, like helices are a very common element in proteins. And I remember a paper on, well, the last element of a helix is going to be one of these 3 amino acids. The one before that's going to be some of, so there's some regularities and human rules that they've cataloged. And you can kind of use that, but ultimately it only works a little bit. It doesn't give you the kind of precision you need to do drug development at all. In terms of things actually that surprised, actually a real surprise came from machine learning. I shouldn't have been surprised, but I was. I mean, there were 2 big surprises. One was sometimes we would have proteins with giant voided cavities in the middle, or a protein that was like C-shaped. And, proteins, the atoms in proteins are really up against each other, right? It's a very dense object. And I said, well, it doesn't look right. But the model was extremely confident. And we'd look in the experimental structure and then immediately realized what would happen. So AlphaFold 2, the original AlphaFold 2, was trained only on single proteins. But often when a protein is solved, sometimes multiple copies of itself will appear, what's called a homomer. So maybe 3 copies actually sometimes densely intertwine with each other to make the actual folded thing is not 1 copy, it's the 3 copies together, a trimer. Or there would be some other protein of a completely different type that it would say wrap around, and they only appear together in the body. And sometimes AlphaFold would realize these patterns and leave these giant voids that look totally wrong, or this spiral which is just floating in air, and I'd be like, well, that's wrong. But it's extraordinarily confident, and then I would find out, oh, it realized that in fact this protein comes in 3 copies, and so this spiral is 1/3 of that, and if you overlay it, it's perfect. So that even though AlphaFold, we didn't tell it about this context, it had learned rules that sometimes there are these geometric patterns which I can explain. I think the other big surprise was actually when we ran AlphaFold across random proteins in humans. We would see some bits that looked beautiful and structured and some really ugly long arcing ribbons. Oh no, that's wrong. And I remember we looked at that, and we wouldn't see this very much when we predicted proteins that were experimentally solved. We said, oh no, our proteins that have been experimentally solved more are special. And actually AlphaFold isn't good on the things we hadn't solved. And then Catherine on the team, a little later that day, looks in this Uniprot, this database of various experimental facts about proteins, which will tell you certain regions that are known, for example, experimentally to be disordered. And she starts to realize that where AlphaFold is making these ridiculous long arcing predictions that can't possibly be correct, and they aren't proteins, it was very low confidence, and those regions were disordered. And what AlphaFold was in fact telling us is this region doesn't have a structure. Kind of implicitly. So what we found out is that the lowest AlphaFold confidence protein was actually pretty much a state-of-the-art predictor of whether a protein was disordered. And so we would find all these things that we kind of knew about proteins, but we didn't feel because disorder doesn't appear in this database of protein structures. We would find all these things out just kind of looking at AlphaFold and being surprised.
Mm-hmm. Amazing, amazing. Now, I'll not ask what its most impactful application is because it has now hundreds of thousands of research works building on it in just about 5 years, which is this is unbelievable. So which one is your favorite?
I think I have 2 favorites. One was this giant protein complex, hundreds of protein chains called the nuclear pore. The nuclear pore is actually kind of the giant gates for the nucleus. The nucleus stores your DNA, right? It's where your nucleic material is, and the rest of the cell is outside the nucleus, and so you need a gatekeeper that decides who can enter and leave and kind of opens and contracts. And I remember thinking, this is enormous. AlphaFold does, it's 1,000 times bigger than what AlphaFold can do. So we, maybe later we'll come up with some machine learning that will help with these kind of problems. And then this paper comes out. The first one I saw was out of the Kosinski and Beck lab saying, we solved the structure of the nuclear pore that we knew something like 30% of before. Now we know 60, 70%, and a lot of the rest is actually disordered. Because we combined very low resolution experimental techniques, cryo-ET, with AlphaFold for the individual pieces and running different AlphaFolds and finding all the little kind of joins and compartments, and then we could finally build the model of the nuclear pore. And in fact, that and some very related papers were a special issue of Science. all about the structure of the nuclear pore, and 3 out of the 4 made huge use of AlphaFold. I remember searching through these papers and maybe 150 mentions of the word AlphaFold in this, in work that we didn't do, that all we did was make the software tool that scientists use to make amazing discoveries. And I just felt like, you know, the Nobel's extraordinary, and now I'm waiting for the Nobel of someone who used AlphaFold and their own creativity to discover the next
Nobel.
thing. Yeah, the second order Nobel is the one that I can't wait for. And I think the other one was people discovered all these uses of AlphaFold that we didn't expect to really work. So they would run thousands and thousands of AlphaFold predictions and just see which one that AlphaFold liked. So the one I really loved, there was a paper on fertilization. How does egg and sperm come together? And there are proteins on egg and there are proteins on sperm that kind of join together and they recognize each other and they start fertilization. But it was known that there was a protein in humans that was missing, that something didn't make sense. And there were actually 2 labs that did this, that took this protein on the egg and 2,000 proteins, every one that appears on the surface of sperm, and just ran 2,000 AlphaFold predictions. And they found one specific protein that AlphaFold thought stuck up against this egg protein. Wow. And then they go to the lab and they say, knock this protein out. And egg and sperm will come together but not start fertilization. They'll make mutations in the individual regions in which these come together, and they'll find out that blocks fertilization. So they've established biochemistry now. This thing they had no idea. They had no idea which of these 2,000 to look at. And AlphaFold said, look at just this one. And sure enough, that was the protein that was essential in this. And I love this notion that we would never do this with experiment. You would never send out 2,000 labs to make 2,000 structures and see which one comes back. That we can do new types of science because of the scale we've achieved.
Yeah. Incredible. Any unexpected use cases?
All right, so one that really surprised me. I can tell you an unexpected weakness and then an unexpected strength of AlphaFold. So the unexpected weakness is if you take a protein and you break it, you do something that's going to cause it to be unstable. Like one very strong rule of proteins is that positively or negatively charged amino acids don't appear in the greasy middle part of a protein, right? They don't like grease. And so aspartic acid is a very small charged amino acid. It doesn't really appear in the center of proteins. So if you take a protein and you mutate one of the inner amino acids to aspartic, AlphaFold won't really change its structure, even though this doesn't make sense. And there's reasons you can explain it, but we say AlphaFold is not extremely point mutation sensitive. It's answering a slightly different question. So we said, okay, that's some future work. And so there are a lot of people who do protein designs, and we're using AlphaFold to check their designs and say which ones, does their design method work? Does it produce sequences that AlphaFold thinks folded to the structure they were trying to make? And I remember thinking, oh, that's probably not going to work because AlphaFold isn't mutation sensitive. It doesn't have a sensitive enough understanding of the interactions. But I was totally wrong about that. And people found that it was actually really good when it came to design proteins at figuring out which ones might work. One paper that came out a few months after AlphaFold said that when designing proteins to bind to each other, they get a 10-fold increase in success rate if they only make the things that AlphaFold thinks binds. Mm-hmm. And it's become really dominant, actually, that AlphaFold filtering is one of the secrets of modern protein design. Even though we were designing a natural protein system, we got kind of this enormous design improvement for free.
Mm-hmm. Now, just to showcase the influence of AlphaFold, in my opinion, let me hold on to my papers for this one to make sure I word this properly.
Oh, yeah.
In 20 years, nearly every person with access to modern healthcare will benefit from a tool, diagnostic, or drug influenced by AlphaFold. What do you think?
I think that's pretty fair. I think that it is now a tool of modern biology. And I will say that there are other tools, like every biological discovery today in some way benefits from DNA sequencing, right? DNA synthesis, right? These are tools that underpin the kind of technology of modern biology. And AlphaFold is very certainly one of those people teach it to grad students, right? It's a standard part of the graduate curriculum. We will learn how to do some things, and I will show you how to use AlphaFold because you will probably use it in your research. And then people make all these discoveries, and these discoveries compound and grow. That's the wonderful part of working in research is that you have this enormous spreading out of the work you do. It's not just, it's wonderful. I think sometimes it's wonderful to be a doctor, to be someone who very definitely and obviously decides the right treatment for a patient and makes someone healthy. But I also love the thought of being a researcher, that I can build a tool that will help 100,000 people, that will help 1 million, that will help 1 billion be healthy in the fullness of time. As kind of, it helps bring forward science. I like to think that AlphaFold maybe made structural biology, which is one of the major fields of biology, 5 or 10% faster, right? And that's extraordinary.
Is it a possibility that, AlphaFold lists, it gives you a confidence score too, not just a prediction. Can it be confidently incorrect?
Yes. So, a very simple analogy. If the weather report says there's a 90% chance of rain today and it doesn't rain, was it wrong? Some people will say yes, but that's not obviously correct. Yes. You're supposed to be wrong 1 time in 10. So we can say AlphaFold's confidence is calibrated. So that's what we can really say, is that 90% chance of being, or really what we say is average accuracy is 0.9 on a certain scale called LDDT. So if our confidence says 0.9, then on average it will be there, but some of them will be very bad. And actually we know a very interesting failure mode of very high confidence. Sometimes it's just wrong, but more commonly, for example, a protein will have 2 structures and AlphaFold will produce one with high confidence, but it won't, but you really wanted the other one. And so its confidence more reflects, does this structure make sense as one state of the protein, but it doesn't necessarily say it's every state of the protein or the one you care about.
All right, let's have a lightning round. I ask you something and try to answer in 1 sentence.
Oh, that's hard for me.
How did AlphaFold 2 improve on the first one?
We did machine learning research at the intersections of protein and ML, not taking ML off the shelf and applying it to proteins.
AlphaFold 3.
We expanded it to do the protein cinematic universe, and we adjusted the architecture to make it work.
AlphaProteo.
It developed new techniques to design more efficiently using AlphaFold and other ideas.
Favorite 2-minute papers episode?
Oh, AlphaFold, it's easy.
Kidding, yes. All right. John, I've learned so much again. Huge honor. Thank you so much.
It was a pleasure. Thank you.